For a century, our sharpest weapons against cancer have been blunt. Surgery, chemotherapy, radiation. All of them harm healthy cells along with the sick ones. The dream has always been a treatment that can tell friend from foe. Now imagine destroying a cancer cell with nothing but sound. A gentle pulse that leaves the healthy cell right next to it completely unharmed. No drugs, no radiation, no incision. Just a vibration in the air, tuned to be lethal to one cell and harmless to its neighbor. It sounds impossible. But a new study suggests it might come down to a single physical difference between the two cells. Not their chemistry, not their genes, but their shape. To see how, we have to start with what sound really is. Sound is not a thing. It is a motion, a wave of pressure, squeezing and stretching whatever it passes through. Picture a long line of people passing a nudge down the row. No single person travels, but the nudge does. That is a sound wave, moving through air, through water, through living flesh. Above about 20 kilohertz, those waves climb beyond human hearing. We call that ultrasound. And two dials decide what it does. Frequency, how fast it vibrates, and intensity, how hard it pushes. Turn intensity high, and ultrasound becomes brute force. It can heat tissue until it burns or shatter a kidney stone. But turn both dials low, and something far gentler happens. It barely warms the tissue. It simply nudges it. This is where our paper lives. Low frequency, low intensity ultrasound. And we have trusted ultrasound for decades. It is how we first see a baby in the womb. Pulses sent in, echoes read back, a picture built from sound. It is safe enough to point at the most fragile life there is. It heals, too. Low-intensity ultrasound is used to coax broken bones to knit back together, the gentle mechanical nudging waking the cells that rebuild them. But then researchers noticed something stranger. At the right low settings, ultrasound could push certain cancer cells into apoptosis, while the healthy cells around them simply carried on. Apoptosis is not damage. It is a decision. Every cell carries a built-in program to die cleanly when it becomes too broken or too dangerous to keep living. Cancer is, in part, a failure of that program. So a tool that could switch it back on, selectively, would be extraordinary. And yet the same wave, the same room. One cell dies on cue. Its neighbor doesn't even flinch. For years, nobody could fully explain why. This is the real mystery. Not that sound can kill a cell, but that it can choose. Why would one identical pulse be deadly to one cell and invisible to another sitting right beside it? Two cells can carry the same genes and the same proteins and still be built differently, like two houses from one blueprint. One sound, one warped. The old guesses looked at chemistry. Different molecules, different genes. This new paper proposes a different answer entirely. The selectivity is mechanical. It comes from the physical architecture inside the cell and the shape that architecture gives it. To understand that, we need to go inside. A cell is not a bag of fluid. It is a structure held in shape by an internal skeleton, the cytoskeleton, made of protein cables called stress fibers, pulled taut like the guy wires of a tent. These cables are bundles of actin, the very same protein that lets your muscles contract. Under tension, they are always pulling, sensing, adjusting, giving the cell its shape and its grip on the world. Because they don't float freely. They anchor to the outer membrane at docking points called focal adhesions, rivets where the cell grips the surface beneath it and pulls itself into shape. In a healthy cell, this scaffolding is orderly. The cables are evenly spaced, the tension balanced, and the membrane is pulled into smooth, regular curves. In a cancer cell, the same machinery is in disarray. Mechanically, cancer cells are known to be different. Softer, more disordered, their tension uneven. The fibers tangle and pull unevenly, and where they tug on the membrane, they crumple it into irregular, jagged curves. Hold that idea, the curve of the membrane, because everything turns on it. That membrane is extraordinary. A double sheet of fat molecules, only 2 molecules thick, yet it is the border that decides what gets in and what stays out. And it is not rigid. It flows, like a soap film, bending and rippling with every force the cell feels. Floating in that film is cholesterol. We think of it as something in our arteries, but inside a membrane it is a tuning knob. It stiffens the sheet, and it decides how the proteins within it cluster together or drift apart. Proteins drift through this fluid like boats on water. Where the lipids pack tightly with cholesterol, they gather into little neighborhoods. And where the membrane bends, those neighborhoods rearrange. Here, shape is not decoration. It is information. So now we have 2 ingredients sitting in the same skin. Its curvature, how sharply it bends, and its cholesterol, how its parts are arranged. The paper's whole argument is about what happens when a force sensor gets caught between them. That sensor has a name, piezo-1, one of the cell's force-feeling channels, a gateway in the membrane that stays shut until it is physically pushed. You feel piezo-1 working right now. The pressure of the floor against your feet, the stretch of your lungs, the flow of blood through your vessels. All of it is read, in part, by these channels. The scientist who discovered them, Ardem Patapoutian, shared the Nobel Prize in 2021. Each one is a curved, 3-bladed dome, like a tiny ship's propeller, only about 28 nanometers across. It bows the membrane around it into a little dome. And here is the key. Piezo-1 doesn't just sense force, it senses curvature. When the membrane around it bends or flattens, the blades flex and the gate opens. When it opens, ions rush through, especially calcium. And calcium is not just a charge, it is a message. A brief pulse of it is normal. But a flood that won't stop is one of the oldest alarm signals in biology, a signal that can tip a cell into self-destruction. So the whole question becomes simple to state. Under that gentle ultrasound, whose piezo one stays open and whose stays quiet? Here is the model the paper builds. In the cancer cell, those jagged, irregular curves near the focal adhesions are exactly the shape piezo one likes. The sensors sit loosely packed in the bent membrane, primed, and easy to open. Watch the difference. On the left, the cancer cell, its membrane already buckled, its piezo one primed. On the right, the healthy cells, smooth and calm. One wave sent through both. When the ultrasound works that already warped membrane, the cancer cell's channels fire and keep firing. Calcium pours in and doesn't let up. The relentless signal pushes the cell over the edge, into apoptosis. The cell is talked into killing itself by its own broken shape. Now the healthy cell, under the very same wave. Its membrane is smoothly, regularly curved. That regular curvature reshuffles the cholesterol, and the cholesterol scatters the piezo one channels apart and damps them down. Barely any calcium gets in. The cell shrugs and lives. Same sound, two shapes, two completely different fates. So the selectivity was never magic, and never really about chemistry. It is geometry. The cancer cell's own internal disorder, the very chaos that helps it grow and spread, becomes the thing that betrays it. Its shape is its weakness, and sound is the key that fits the flaw. It reframes the whole disease. We usually fight cancer's chemistry. This says, look at its form. The disorder that makes a tumor aggressive might be the very handle by which we finally grip it. Now, the honest part. This is a theoretical model built on top of earlier experiments. Not a finished therapy sitting in a clinic. It is a framework, a mechanism you can test and argue with and try to break. But that is exactly what makes it powerful. If a cell's vulnerability is its shape, then one day we might design ultrasound that targets cancer by its geometry instead of its chemistry, sparing the healthy tissue woven right through it because that tissue is simply shaped differently. A cell's own broken architecture turned against it by nothing more than sound. This is Paperscape, where research becomes a world.